I'm Trudi Schupbach. I'm a Howard Hughes investigator and professor of molecular biology at Princeton University. In the first part of my talk, I have told you about establishment of pattern polarity during Drosophila oogenesis. I've also told you that there is a key molecule, Gurken, that gets localized during oogenesis. And in the second part of my talk, I would like to introduce you to some of the experiments that we are doing in order to understand this Gurken localization process in more detail. Just to remind you, however, I'll show you again the picture of a Drosophila ovary. At the top, you have the youngest egg chambers. They develop continuously down to the older stages. Pattern and polarity can already be seen at later stages of oogenesis in the egg chambers. And in particular, we will concentrate here on the dorsal-ventral asymmetry. And that is predictable at this stage because the oocyte nucleus is localized to one particular corner of the developing egg chamber, and whichever side of the egg chamber it's closest to, that's going to be the future dorsal side of both the egg and the embryo. The Gurken molecule, which encodes a signaling molecule, is produced by the germline and accumulates in close vicinity to the oocyte nucleus. You can see here the Gurken RNA surrounding the oocyte nucleus, and this RNA then gives rise to the protein, the Gurken protein, which is a transmembrane protein and accumulates to higher level in the oocyte membrane, but again to a very restricted part of the oocyte membrane, just in the dorsal anterior corner. The Gurken molecule encodes a signaling molecule of the TGF-alpha-like family of growth factors. It can be released from the oocyte and then binds and activates the Drosophila EGF receptor, which is a large transmembrane receptor that sits in the membrane of the follicle cells. And upon receiving binding the Gurken signal, the EGF receptor will activate its internal tyrosine kinase domain, and that leads eventually to the activation and repression of subsets of genes in the follicle cells and induces the follicle cells to become dorsal follicle cells. In order to understand how the Gurken RNA molecule gets localized to this particular place within the oocyte, we used again the mutagenesis approach because that allows us to see without bias what type of genes might be involved in getting the RNA to be accumulating just in this one particular place during oogenesis. So in mutagenesis experiments that either we or other people did, we found a number of genes in the Drosophila genome that, when mutant, resulted in a mislocalization of the Gurken RNA. In particular, there is a group of genes that has the following phenotype. In these genes, the Gurken RNA, rather than being restricted to just one side of the egg chamber, is found all around the anterior circumference of the oocyte. You might appreciate this better in a cross-section shown in this picture here. You can see in wild type, the Gurken RNA is just localized sort of like a cap above the oocyte nucleus on the dorsal side of the egg chamber. In contrast, in the mutants here, you find the RNA all around the anterior circumference. Therefore, the genes that are mutated in these various mutant lines must be involved somehow in getting the RNA to be just localized to one side of the egg chamber. And so, of course, we're very interested in what these genes encode. The ultimate result of this mislocalization is a dorsalized egg and embryo. This is because when the RNA is not localized to just one side of the egg chamber, but is found all around the anterior circumference, then we also see the Gurken protein all around the anterior circumference. And now follicle cells from all sides, both the dorsal and the ventral sides, will see high levels of this Gurken protein, think that they are dorsal, and start migrating and producing dorsal appendage material. The first gene in this class that we looked at in more detail is the gene Squid. Squid was originally cloned by 2 different laboratories simultaneously. It encodes a protein that can bind RNAs, and it can also shuttle in and out of nuclei. Amanda Norvell, a former postdoc in the lab, looked at Squid in more detail, and she found that Squid can indeed bind the Gurken RNA directly. She also discovered that Squid is not only required for the localization of the RNA, but it's also required for the translational repression of the RNA before it's localized. Many localized RNAs are initially translationally repressed. The cell will do this because the whole goal of RNA localization is to get the RNA to the place where the protein should be localized. And so the RNA should not already be translated during its journey to that location. And for that reason, very often these localized RNAs are associated with repressive complexes that keep them from being translated while they're still being transported. This seems also to be true for the Gurken RNA, and Squid is an important factor in organizing such a translational repression. Now, Squid is of course not doing everything by itself, localizing and translational repression, and so we wanted to see what other proteins would associate with Squid and help in the function of Gurken localization and translational regulation. So 2 graduate students in the lab, Jennifer Goodrich and Nicole Klaus, undertook some screens to find proteins that would interact with Squid. They used different methods. One method is to go to yeast and perform what we call a yeast-to-hybrid screen, where we transform Drosophila proteins into yeast. And the system is set up in a way that if 2 of these Drosophila proteins bind to each other, the yeast will be able to produce a blue color. So in this way, Jennifer and Nicole found this gene HRB27C, it's also sometimes called HRP48, which can associate with Squid. They also verified this by doing more directed physical interaction tests, for instance, using IP methods to immunoprecipitate with the help of Squid, the complex, and then testing whether HRB27C could also be seen in this complex. Now, showing that 2 proteins interact physically doesn't necessarily tell us whether they really also functionally act in the same process. And so Jennifer Goodrich tested mutations in HRB27C to see whether they really also affected Gurken localization. This wasn't so straightforward in this case because mutations in this gene are actually lethal. That means we can't just simply take homozygous females and check their eggs, but we have to use a more complicated way of making mosaics where the females in general are heterozygous for the mutation and therefore survive, but then using mitotic recombination we can create homozygous germ cells and then test what the effect is of losing this gene now in the germline. Performing such a test, Jennifer found that indeed the eggs produced by such germline clones were also dorsalized, and importantly, the Gurken RNA was not localized correctly in these mutant egg chambers. HRB27C has a number of other phenotypes. You can see these eggs are smaller. We also see abnormalities in the nerve cells. So this gene has multiple functions during oogenesis, but importantly, it does appear to create the same mutant phenotype as mutations in squid, and therefore seems indeed to interact with squid in this particular process of Gurken RNA localization and translational control. Performing more such screens and tests, we came up with the following cartoon interpretation of our findings. The Gurken RNA is transcribed in a nucleus, and presumably in the nucleus it will already interact with genes like squid and HRB27C, which accumulate in the nucleus and can bind to RNAs as they are being transcribed and spliced. These factors then seem to leave the nucleus with the RNA, and in the cytoplasm it appears that squid and HRB27C can organize a complex on the Gurken RNA that allows the RNA to move, to associate with the localization machinery, but also to keep the RNA translationally repressed. Once the RNA has reached its destination, there seems to be some sort of anchoring mechanism that anchors the localized RNA in this place. And this also results somehow in a reorganization of the protein complexes that are associated with Gurken, so that the repressors, the translational repressors, leave the RNA and open it up for translation, so that now in this localized state the Gurken protein can be produced. Now, we have, of course, many questions here. For instance, what is this anchor that anchors the Gurken protein in this dorsal anterior corner? We have a candidate protein that we think might be involved in this anchoring, and that is the protein encoded by a gene called ENCORE. ENCORE is a gene that also has multiple functions during oogenesis. But at this particular stage in oogenesis, it seems to be required for high levels of Gurken protein accumulation. In the ENCORE mutants, you can see the Gurken RNA is somewhat localized. There is more RNA on the dorsal side than on the ventral side, but it's not very tightly localized to the dorsal anterior corner. And then very dramatically, the amount of Gurken protein that we see at this stage of oogenesis is very much reduced. Because there is not much Gurken protein in these eggs, this results then in a ventralized egg and embryo phenotype, so that instead of having two populations of dorsal follicle cells that produce dorsal appendages, we lose the dorsal midline and we have only a reduced population, single population of follicle cells that will migrate and form dorsal appendages. And the embryos that develop inside these eggs are also ventralized. When Cheryl van Buskirk, another graduate student in the lab, made an antibody to the ENCORE protein, we found that this protein also accumulates in the dorsal anterior corner to higher levels. You can see here in a cross-section ENCORE protein around the oocyte nucleus at higher levels than you find it on the ventral side. The ENCORE protein is not quite as tightly localized as, say, the Gurken RNA, which is shown here for your reference in this panel, but nevertheless, it appears that ENCORE shows an asymmetry that makes it a candidate for being such an anchor of the Gurken RNA. Another finding about ENCORE is that it interacts with a polyA binding protein. PolyA binding proteins are usually required to upregulate translation in cells, and we found that ENCORE seems to directly interact with such a polyA binding protein. Putting all these results together then into a scheme, we now think that once the Gurken RNA reaches its destination in the dorsal anterior corner, ENCORE might help to anchor the Gurken RNA, and it may also be involved in upregulating translation of Gurken at this stage through its interaction with a polyA binding protein. Of course, there are still many more questions that you would like to answer. We would like to understand how the Gurken RNA complex is actually moved through the cytoplasm. This cartoon here was obtained by either doing genetic experiments or by checking for direct physical interactions. But a lot of it is based on interpretation of these results. And what we really like to do in the long run is to visualize the RNA and the proteins in real-life oocytes and ask, are these proteins really all together sitting on the RNA as it's moving? Who comes on when? Who leaves the complex where? And what happens in a more dynamic and biophysically described manner? As a first step in this direction, we have started a collaboration with my colleague Elizabeth Gaddis in our department at Princeton University to visualize the Gurken RNA localization process live. The methods that we're using here is to try and label the Gurken RNA so that we can see it live in the microscope. The methodology for such messenger RNA labelings have initially been worked out by Rob Singer and his colleagues in yeast, and then have been adapted for Drosophila by Liz Gavis and her students. The system consists of two parts. For one takes the RNA one wants to study, Gurken in our case, and inserts some stem loops into this RNA in a place where it doesn't interfere with the function of the RNA. These stem loops are derived from an RNA virus, and there is a viral coat protein that has a very high binding affinity to these stem loops. So then the second part of the labeling system involves the viral coat protein now fused to GFP, which is co-expressed with the marked RNA. And then when both of these parts are together in the same oocyte, the GFP-marked coat protein will bind to the stem loops and thus mark us the live RNA so that we can see under the microscope where it is. Angela Jaramillo, postdoc in the lab, used this system and made the Gurken constructs and was able to see indeed that we could visualize the Gurken mRNA accumulating either at the posterior end of the egg chamber or at the dorsal anterior corner later in oogenesis in live imaging. Angela then used the system to ask a question about the dynamics of the Gurken localization process. And for this, she used a technique called fluorescence recovery after photobleaching. What this involves is to take a laser and make illuminate a small portion of the localized RNA and make those molecules there that are in the laser beam dark. Now, if the molecules are stably anchored, they will just stay there, and therefore there will be a dark hole in this spot persisting. On the other hand, if the RNA molecules are not stably anchored in this position, but are constantly exchanged with new molecules coming from the cytoplasm, then this initial dark spot will over time recover fluorescence. And so that's what Angela did, and then she measured what would happen after she used the laser to bleach the molecules in particular spots. So I'm going to show you two movies. The first movie here was taken at an early stage in oogenesis when the Gurken RNA is localized to the posterior pole. And you will then see for a short amount of time, as you could just see now, a dark spot where the RNA was bleached. But you also will probably notice that very quickly the spot was filled in and the fluorescent was recovered. We will now compare this to the second movie, which was taken at a later stage, stage 9 of oogenesis. And if we run the movie now, you'll see again the dark spot appearing here, but this time it's not being filled in very rapidly. It remains there during the duration of the time that we took this movie, about half an hour, and was not filled in, telling us therefore that at this stage the molecules are much less dynamic than in the early stage. Angela, of course, measured the fluorescence very carefully, so she could show that after the initial drop in fluorescence gets recovered, and that this recovery changes over time. So in summary, when she summarized all her results, she found that over time there is a decrease in the recovery of fluorescence from young to old stages, and in particular there is also a big jump in recovery time from the younger to the older stages. All of this tells us that the Gurken RNA seems to be localized at 2 different stages by 2 different mechanisms. Early, at the posterior ends, the Gurken RNA seems to be very dynamic. This recovery that we saw, this rapid recovery of fluorescence, is consistent with a continuous exchange of new molecules coming in from the cytoplasm. And it might therefore reflect constant transport. In contrast, at the later stage, the Gurken RNA seemed much more static. It was not able to recover very quickly or very well at all. Therefore, we would conclude that at this later stage, there is an additional mechanism that really anchors the RNA. And remember now, in the beginning of my talk, I said that we require these molecules, Squid, HRB27C, Encore, mostly at this stage of oogenesis. So they seem then to be particularly important to get the RNA to this anchored stage, which after all lasts for a long time, but the RNA needs to be present over quite a long time in oogenesis.